Before we get into the paper, we first need to understand the cell mechanisms that Toda et al. Took advantage of. First, they take advantage of Notch signaling. In this mechanism, a transmembrane receptor called Notch is activated by a transmembrane ligand from a neighboring cell. Upon activation, a protease cleaves off the intracellular domain of the Notch receptor, which translocates to the nucleus and turns on transcription for certain genes. Another cell mechanism Toda et al. Utilizes is cadherins, which provides an adhesive bridge between cells, allowing them to stick together. In Toda et al., they use two different properties of cadherins to get differential adhesion. First, cadherins have homophilic adhesion, meaning cells with the same type of cadherin will adhere together, i.e., cells with N-cadherin will stick with other cells with N-cadherin. Additionally, cells with high expression of cadherin adhere differently to cells with lower levels of cadherin. Mixing cells with high and low expression of cadherins, the cells will come together to have a high cadherin expression core and low expression outer layer. In Toda et al., they attempt to engineer cells that self-organize into organized multicellular structures. They do this using a synthetic notch or syn-notch receptor that recognizes different ligands that drive the expression of different genes. In the first experiment, they use two different cell types. Cell type A expresses blue fluorescent protein and the CD19 ligand, while cell type B expresses an anti-CD19 syn-notch receptor. When the anti-CD19 receptor is activated, it drives the expression of E-cadherin and green fluorescent protein. The resulting structure is a ball with a green core, with high adherence because of E-cadherin and a blue outer layer. Toda et al. Then tries to have organized structures with one cell type. This cell expresses CD19 ligand fused with M-cherry and anti-CD19 syn-notch. When the anti-CD19 syn-notch is activated, it drives expression of E-cadherin and GFP fused with a TET repressor that represses CD19 fused with M-cherry. If a cell's CD19 ligand interacts with another cell's syn-notch, then it represses the syn-notch receptor. The resulting structure will be a green core because of E-cadherin expression and a red outer layer. Toda et al. Also creates many asymmetric structures using the same sort of logic as before, also taking advantage of different types of cadherin. Overall, however, Toda et al. Proves that self-organizing structures can be made, opening up possibilities of forming synthetic tissues and more.